So I was mentioning that excellent work is done by, in the CAR T field by two pioneers like Carl June and Steve Rosenberg. And both of these. So right now regarding CAR T cell, although if you see the geographical disparity, it's mainly in the developed country, especially in United States or Europe, as well as in our neighboring country like China. However, unfortunately, what you see in certain zones are completely empty, including India and South Africa, as well as the Middle East, right? We don't see anything there. And although the field is progressing very well, especially if you see in 2010, there were only 3 trials. Now, probably more than 1,000 trials if you combine all kind of CAR T uh, targets, right? And how the CAR T platform works, but in very brief, generally right now, the current CAR T platform is mainly the autologous platform where you get the immune cells via apheresis from the patient, from the individual patients, and then you send this apheresis sample to the laboratory for the genetic engineering and for the CAR T cell expansion. And once we have enough CAR T dose, they are reinfused into the patients with certain chemotherapy, especially the lymphodepletion therapy. And that's how at present the current CAR T is done. The model of CAR T manufacturing at this moment, especially by the big pharma like Novartis, the way they are doing it, they take the T cells from the patient, and these T cells then are shipped to a larger global manufacturing facility. And in this particular facility, which is a currently in one or two in one country, they manufacture the cells and then ship these cells back to the clinical site. And now this is currently working well, but it's a very expensive model. So one of the things what we can think of, can you manufacture the CAR T locally? And then probably the logistics are easy. And this model can be now tried in the LMIC and country like us, right? So CAR T is very successful in certain malignancies, especially the B cell malignancies. And at the present, only CD19 trial or CD19 product is in the market. And if you see the CD19 expression, it is start expressing from the pro B cell stage once the stem cells is committed to the B cell lineage, especially from the pro B cell, until the plasma cell. Most of this B developmental stage of multiple B cell, they all express CD19. And that's the reason why CD19 target is excellent target, working very well for the eliminating all these kind of malignant cells. Okay. However, in multiple myeloma, CD19 expression is controversial because there are few patients where CD19 CAR T worked, but it's still controversy whether plasma cells really express CD19, whether multiple myeloma is a good target for these particular CAR T. Other CAR T are in the field are CD22 or CD20, which are still in the investigational stage. Okay. So if you just see the, in summary, the clinical trial or the institutions which are doing the clinical trial of CAR T or where CAR T is done, mainly again, the developed country as well as some in China. And many of these trials or the patients, they are treated ALL, NHL, or the CLL. Right. Right now, CAR T seems very efficacious, not just for the short term. They can provide you the long term uh, remission. That means it is intent to cure rather than, generally in cancer field, you listen a lot of remission, but this particular type of treatment you can think of intent to cure. So can the cancer can be cured, and this is one of the good model where this girl Emily Whitehead is treated. She was one of the first patient who was treated, and until today she is fine, she's cancer free, and now you can clearly see she's a young adult now. right? CAR-T platform is making a lot of noise, probably not just good for the hematological malignancies, but now it is moving towards other malignancies. In fact, there are some investigational studies where they are showing good response for solid tumor like breast, prostate, and glioblastomas. But not just oncology, it is even going beyond oncology. So Nature paper just last year came from the UPenn group showing that can you treat cardiac fibrosis with the engineered CAR T cell. Yesterday, there was first patient dosed for CAR M. That means not just CAR T, there's CAR macrophages they made and dose first patient in University of Pennsylvania, right? So a lot of good progress is made. But when you think about LMIC, these are the figures which are exorbitant figure, especially for the affordability point of view in LMIC, which actually a major concern. So all these drugs they are approved in a developed world as well as some in Asia as well, in all the continent, but they cost a lot, which is a very big concern in our countries, right? In India, in Indian currency, it goes up to 4 crore only for the drug. Then you have to pay additionally for the clinical care. So that brings us to a big question: should we have something indigenous which is more affordable to our patients and can help in the cure? So what are the CAR-T challenges, especially in terms of the LMIC perspective or Indian perspective as well? So we have now cancer incidence are very high. Given our economy is doing well or growing faster, cancer incidences generally are increasing. Availability of the CAR-T not available even at any price in Indian market, not just for the poor, even for rich, it is not available. In our country, there's a lot of technical and clinical training, which is required, is lacking right now because it's a complex process. As I mentioned already, the cost and affordability is a huge challenge. In addition, in LMIC, infrastructure is a challenge because you need very good facilities to manufacture these cells. And so along with it, the regulatories, because we need to have the regulatory infrastructure in place to perform these complex studies. So can DIY of CAR-T is a good model? So probably yes, but we have to try and validate it. So can we have something like this model where the innovator lab do all the R&D work, provide to the pharma, and pharma then provide the vector and other support, and at the clinical site we can make the cells locally. So this model could work, but it still need to be validated, right? Current CAR-T, if you see where is the major cost, so you see that current cost is almost $500,000. Manufacturing cost, depending on the site, it costs somewhere between $75,000 to even $100,000. But if you see notice in this graph, what is very interesting is that 60% of the manufacturing cost goes to the skilled manpower. That means skilled manpower is costing more than half of the cost. And if you think about country like us, especially India, here we have skilled manpower. Only thing we need the training. If once we train our clinicians or the biotechnologists and scientists, we can significantly bring the cost down, because 60% of the cost of manufacturing goes in the manufacturing. So that could be an opportunity instead of the challenge. So what we need to do this, right? So what we need is that, again, scientists and clinicians should talk more, and collaboration and partnership, and of course, getting a better infrastructure, invest in infrastructure. These are the two very key to bring the CAR-T. Some automation can also help, because to remove or to eliminate the center to center variability, some automation can help. There's no complete automation right now, but there are machines or there's semi-automation available, and there are multiple companies are trying that, such as GE Healthcare. They brought multiple individual equipment, and they're all integrated. So they're semi-integrated, where you can do each process at one time and can get the CAR-T cells. Similarly, another semi-automated system, another came by the Miltenyi Biotech called Prodigy. Again, some processes you can do in automation here as well. Terumo is coming up again with the many good platform. And similarly, there is semi kind of automation system here in the manual process as well. So what would be better? We do not know yet, but when we started our project almost 6 to 7 years ago, we thought our objective should be that can we reduce the cost to 70 to 90%? Can we bring cost from $375 or $400,000 to only $15 to $20,000, say, per patient, which might be beneficial because that's something like this is the cost of the bone marrow transplant in our country, which can be affordable to at least some section of the society, if not all. Right. So our idea was that can we get mastered in the this complex technology, complex manufacturing process. And in fact, in CAR-T, just for LMIC people, it's not just the manufacturing. You have to do quality control because this drug can be very, very toxic. So you have to ensure that safety issues are taken care of. For that, you have a big quality control assays to be performed. Our another idea was, can we develop the novel product which are for hematological malignancies as well as for solid tumors. So with our team at IIT Bombay and Tata Hospital, both of us in collaboration with the National Cancer Institute, so Banavali sir, Dr. Gaurav sir, and Nirali and Terry and Steve Heifel, we all made a team and we wanted to work together to bring the CAR-T in India as well as bring the CAR-T affordable to our patient. Okay, so so now I will go brief a little bit about our work, but if you want to read more about our work, it is already published in Molecular Cancer Therapeutics and 2 weeks ago. It is online now. So just in brief, we designed the product, we have the patent on this, and this is a CD19 CAR with 41BB as a co-stimulatory domain. Okay, so we performed the preclinical study, extensive preclinical study, efficacy as well toxicity, which shows that this product or this anti-CD19 CAR T cells, they're efficacious. If you see on the right hand side, these all the mice, they are treated with anti-CD19 CAR T, and the other one on the left, which are control, they are not treated by the CAR T cells, the untreated group, and right, you have treated group. You clearly see that on the mice that were treated with this anti-CD19 CAR, they do not have the tumor, or just very few have very little or negligible tumor burden, indicating that these CAR T are efficacious. Okay, so in summary, what we have is unique CAR T now. So a humanized anti-CD19 construct. Our gene delivery system is third generation lentiviral vector. They are tested extensively in the ex vivo setting, not yet in the clinic, but their in vivo efficacy in the animal model is great. They are less toxic. And in the we also did CGMP feasibility with few relapse and refractory BLL patients, which are recruited at Tata Hospital by Dr. Gaurav Narula sir. And this work is also been patented, and this is most likely going to be first to clinic in our country very soon. So in brief, LMIC, yes, DIY model might be suitable, but clinical testing and validation needed. So this need to be validated whether it will work or not. So long way to go in that regard. Regarding our data on CAR-T, we are most likely going very soon to clinical trial. And then ultimately, we have to bring this technology to save many patient life, including children as well as adult, right? And hopefully we are moving towards clinical trial. Regarding CAR-T, it's still a long way to go, despite this being a very expensive and very hot field. If you see the long-term data started coming, if you see on the right-hand side, all these trials, what you are looking at, despite such an expensive treatment, half of the patients almost relapse. So long-term benefit is only in half of the patients. So of course you need better CAR in future, but at least we can achieve half of the patients under remission for long-term, which is still a very good news for majority of the patients. Long way to go still though. So with this, I will end here with thanks to my team. So I work in the immunology area, and half of the lab works in CAR-T technology development, and other half works in the T-cell cytokines and inflammation and their role in cancer. So majority of the work I presented today is done by Alka, Atharva, Sweetie, Ankesh, and Sarvari Ghosh. And Afreen and Alia, they supported with the quality control assays, etc. So thank you so much. And also, I would like to thank our collaborator and our partner, Dr. Gaurav Narula sir, Hasmukh, Dr. Banavli sir, Dr. Chetan Dhamne, and Dr. Naveen Khatri and Dr. Meenal Pujari. And from NIH, Terry, as well as Nirali Shah and Stephen Heifel, who helped us a lot extensively in developing all this. And without their help, it wouldn't have been working so far. And we have a lot of support for a lot of collaboration from other colleague in the department as well as my institute. And funding, majority of the CAR-T funding came from our intramural funds as well as from the Tata Trust and Vadvani Foundation. Thank you so much.